Live.
All right, on your mark, get set, go.
All right, good morning, everybody. My name is Caroline Lawson. Thank you for coming out today. I chose to do my cell communication presentation and research on holoprosencephaly. So what is holoprosencephaly? So on a broad spectrum, holoprosencephaly is a cell communication disorder that affects the embryonic development of the brain. You'll see it abbreviated on my board and on the board up here as HPE. So let's break down the word. As you see, I have it separated up here, and that's for a reason, because the root, like the prefix of the word holo, is whole, and that's in Latin or Greek, and prosencephalon is the anterior division of the embryonic neural tube that develops eventually into the cerebrum. Why did I choose this disorder? I actually don't have any connections to holoprosencephaly. It is a very rare disorder, which I'll talk about later. But I wanted to do something that I've never heard of before, and probably none of you have ever heard of it before. And it was a rare disorder that I figured I could learn a lot about, and I've really enjoyed researching it. Each and every one of your brains has a right and a left hemisphere. Those are further divided into the frontal, parietal, temporal, and occipital lobes. The right and the left hemisphere stay in contact with each other by the corpus callosum. It is basically a ball of tissue in between your right and left hemispheres, a ball of nerves that helps them communicate with each other to carry out just normal functions of the brain. The midline down the middle is called the interhemispheric fissure. It's the line that you see between every brain if you see a model of a brain. Somebody with holoprosencephaly, though, has a different brain structure. So, on more technical terms, holoprosencephaly occurs when the brain is not separated into two full hemispheres during embryonic development. This usually occurs between the second and third, sometimes fourth week of embryonic development, as that is when the prosencephalon forms. And it often happens before the mother even knows she's pregnant. So we can see it here. Here's the prosencephalon during the 3 to 4 week embryo development. That develops into the telencephalon and the diencephalon, which is the cerebrum, the eye cup. When this doesn't form correctly, that's when holoprosencephaly occurs. So you've heard that holoprosencephaly is a cell communication disease. What is a cell communication disease? Well, cell communication is the way that cells talk to each other almost, the way that they communicate with each other and carry out different processes. This can happen through signaling between either local or long distance, local being between neighboring cells through the extracellular matrix, which is what holoprosencephaly is, since it's all in the brain, and then long distance signaling would be through the bloodstream to different organs, different parts of the body. There are 3 stages of cell communication. What are they? They are reception, transduction, and response. So reception occurs when a signaling molecule from the specialized cell is sent out. It is called a ligand. The ligand goes to a specific protein on the target cell that it is going to, and the receptor binds to the ligand. This causes a bunch of signals, intracellular signals inside of the cell to be released, and almost a cascade effect, a domino effect, called transduction, to carry out a response, which could be gene activation, expression of a gene, other cellular processes, growth, whatever it's supposed to be. In my case, it's supposed to be gene expression to cause the midline of the brain. What does that look like? The specialized cell would send out the ligand here. It would bond to this receptor, causing the chemical messengers to relay the signal and the response. My pathway that I focused on was the sonic hedgehog pathway. The sonic hedgehog pathway is actually known as the master midliner. It is what controls the brain, the face, the separation of the brain. That's why it's known as the master midliner. So how that looks in cell communication is the sonic hedgehog protein, the ligand from the specialized cell, is sent out and activated with patch 1. Patch 1 is a receptor protein. It holds on to the sonic hedgehog ligand. Patch 1 is actually also an inhibitor for smoothen. So smoothen is another protein. Whenever the activated sonic hedgehog binds to patch 1, that is when smoothen is released, there's GLI 1, 2, and 3. Those are the dominating cascade effect of transduction. Those are the intracellular signals that go into the nucleus, causing the gene activation of the midline to split. Whenever the sonic hedgehog protein is mutated, that is when holoprosencephaly occurs. So since it's mutated, it can't bind to patch 1, that can't really smooth it, can't do anything. So the problem actually occurs in reception at the very beginning. Because it cannot bind, nothing else can happen. The mutation of my disease is located on chromosome 7. So at the very end of the long arm, 7q36.3, that is where the sonic hedgehog gene is located. So whenever there's a deletion, a mutation, a duplication, whatever it might be, on the end of the long arm of chromosome 7, holoprosencephaly results. Some history of holoprosencephaly. So holoprosencephaly has actually been around for centuries. It's noted in the 17th century that it was not called holoprosencephaly yet. There were documents of facial anomalies that are the same as holoprosencephaly today, and it wasn't actually until the early 1900s that the brain was discovered, that the separation of the brain was found out that it didn't separate. And then in the mid-20th century, Dorothy DeMeyer actually did research, and she showed that the facial features actually were the result of the midline of the brain not forming. And it helped with modern understanding of holoprosencephaly. Later researchers identified environmental risks like gastrointestinal diabetes and alcohol exposure actually can affect holoprosencephaly. And today, improved ultrasound imaging and X-rays, as we know, have revealed HPE and when it occurs in pregnancy. Some symptoms. So actually about 1 in 6 babies with holoprosencephaly have hydrocephalus. Hydrocephalus, you might have heard of before, it is fluid on the brain. A shunt can be placed to drain that fluid. Babies with hydrocephaly also can have cleft lip or cleft palate just as a result of the improper brain and face formation. They can do facial, plastic surgery on the face to help with that, and that helps with breathing, swallowing, speaking, other stuff like that. And then unfortunately, some babies in the most severe cases of holoprosencephaly have cyclopia, which would be a one central eye, and unfortunately these babies are incompatible with life. Other symptoms: epilepsy, other seizures, difficulty with swallowing, breathing, gastrointestinal problems due to the improper movement of foods. Improper formation of the pituitary gland causes hormonal imbalances, so parents have to look out for stuff like that. A small or large head. I've seen a lot of babies with hydrocephaly who are living. They actually do have a small or a large head. Other facial deformities, and then of course there's intellectual disability with the improper formation of the brain. Treatments: anti-epileptic medications for the epilepsy and seizures. A lot of holoprosencephaly babies deal with seizures. Physical and occupational therapy with everyday tasks, to help with everyday tasks. Speech therapy if they can speak, and then the shunt and the facial surgery like we talked about. There are 3 different types of holoprosencephaly: lobar, semi-lobar, and allobar. Some people pronounce it as alobar. They all range in severity, the least severe being lobar. As you see, I also have this on my board if you want to look at it later. Lobar, the brain is mostly separated. There just might be some parts missing of the interhemispheric fissure, but most of the brain is separated. There might be some slight intellectual disabilities and some slight facial problems with this. Semi-lobar is basically the brain is a U shape. It is fused together in the front, but the back part of the brain is still separated. There are a lot more deaths with this case and a lot more stronger symptoms. And then alobar, unfortunately, you don't see a lot of people live with alobar holoprosencephaly, but it's not impossible. Babies actually live with this, and you could live with this to adolescence or adulthood even, though it is a lot more uncommon. And that's where the cyclopia comes, not in all babies, but the more severe symptoms. And if you want to see that on an actual brain, this is what it would look like lobar, semi-lobar, and alobar. Some facts: holoprosencephaly is actually very rare. It occurs in about every 8 to 10,000 births. This could be live birth or stillbirth, unfortunately. But it's actually a misconception that it is as rare as it is, because about 1 in every 250 embryos have holoprosencephaly, but it results in a miscarriage. It's mainly caused by genetics, but as we talked about, the maternal diabetes or alcohol exposure can cause a higher risk factor, and about 50% of cases are caused by chromosomal errors, and then the other 50% would be with risk factors and a mix. The Sonic Hedgehog gene was named by Robert Riddle after the Sonic Hedgehog, the video game character. He saw it on a magazine actually, and this gene was seen in fruit flies with a spiky appearance in embryonic development, which is just a cool fact. That was the gene that we talked about that messes up that pathway. And there's my works cited. Thank you.
There you go.